my co-chair, Gregory LaMonte from UCSD, and he's going to tell us about novel host biomarkers in liver stage plasmodium infection. Thanks, Greg.
All right, so it's now going to be time for something almost completely different, but we'll go with it. Hello, my name's Greg LaMonte, and I'm going to talk about some of our work trying to understand sort of the liver stage of infection and host pathogen interactions, specifically during the liver stage. And at this point, I think everyone's very familiar with the importance of the liver stage of infection for combating malaria, in particular due to its ability to being the site of malaria prophylaxis, there being reduced parasite burden so that there's reduced risk of drug resistance, and of course, as was discussed repeatedly in this meeting, sort of the dormant stage of infection or hypnozoites, which will be critical to be combated moving forward. And so in my work and in the Winslow lab, we've been trying to use systems biology-based approaches using multiple parallel lines of evidence to try and understand how the parasite actually develops within the liver cell and sort of if we can identify various markers and factors involved in infection. And for today, I'm just going to talk about a pilot transcriptomic study we did using dual RNA sequencing of infected host hepatocyte cell lines. And our goal with this was actually to identify host cell biomarkers or factors expressed by the host cell that are involved in parasite infection, because a lot of people have looked for parasite markers, but we felt there was still a fair amount that we don't understand about the host cell response to this. And why most everyone's familiar with why biomarkers are important, is they'll be critical for identifying either asymptomatic carriers or dormant infections in terms of people who will require treatment. But why we really wanted to focus on a host biomarker of infection is most of the current biomarkers are actually based on parasite proteins. And so to an extent, these are all largely species specific. And so in the best case scenario, if we could come up with a host biomarker that would identify when someone had a plasmodium liver stage infection, this marker could in theory be species independent. But even at sort of the most basic case, we were thinking for sort of an experimental system, if you could identify host factors that had their regulation changed during malaria infection, you could design a series of molecular tools and cell lines that would enable you to sort of perform a lot more in-depth experiments in the plasmodium species that aren't currently genetically manipulatable. For example, you could make a hepatocyte cell line that would self-report when it was infected, and you wouldn't have to modify the parasite because you would be modifying the host cell instead. Oh, where do I point? There we go. And so just to say how we went about this, we used a fairly straightforward system using Plasmodium berghei parasites with expressed GFP, and using fluorescence, there we go. Using fluorescence cell sorting, we sorted into GFP positive or infected and GFP negative uninfected cell lines. And similar studies these have been done, but they've sort of only focused on one cell line. And we wanted to sort of specifically look for markers for the host that were conserved across independent of cell context. So we performed this study in 3 independent cell lines, HC04, HUH751, and HepG2 cells. And we collected RNA right at the point of infection, 0 hours, 24 hours, so halfway through the Plasmodium berghei infection, and sort of late stage in the infection at 48 hours. And just doing sort of a standard unsupervised clustering of this, what you'll notice first is that most of the changes, and I apologize, it's sort of washed out. So the first group here is all HUH751 cells, and then HC04, and then HepG2. And what we found is that the vast majority of RNA changes when the host cells were infected were cell line specific, so that one cell line had a very distinct response from all of the other cell lines. But we did see a small group of 42 genes that were actually predictive of infection across all 3 cell lines. And these had a variety of pathways that they were involved in, including histone remodeling, Golgi transport, nuclear hormone signaling, and a downregulation of DNA replication. But within these, the most predictive gene within that cluster of 42 genes was actually a mucosal immunity protein, mucin 13, which is what I'll sort of focus on for the rest of my talk. And I'll point out that in the next session, the late breaking talks, the second most predictive gene was a nuclear hormone receptor, NR4A3, which another member of our lab will speak of. And so mucin 13, as I said, is a transmembrane mucosal immunity protein. It's about 50 kilodaltons in length. It has extensive glycosylation on the extracellular domain, a transmembrane domain, and several EGF2 and 3 signaling domains. it's been shown to be upregulated in certain cancers and in response to Helicobacter pylori infection. And so before we went too deeply into this gene, we just wanted to confirm that it was in fact upregulated during malaria parasite infection. And so we did that via real-time PCR with independent infections of the same 3 cell lines and primary human hepatocytes. And we see about a 50-fold upregulation of mucin 13 transcript levels. And then by Western blot, you see an almost complete upregulation. It's almost undetectable, and it's a little washed out. There is a faint band there. But you actually see a tremendous upregulation of the protein mucin 13. And so we were very curious about how mucosal immunity protein might be interacting with the parasite. And so we wanted to look into how it was, where it was sort of expressed within the cell when it was infected, and whether it was how it was co-localizing with the parasite. And so we performed immunofluorescence. And what you can see here, and it's a little washed out, is you see a little bit of host cell signal, but in general, in infected cells, the mucin 13 is actually almost completely co-localized with the parasite, suggesting that it might actually be within the parasite plasma membrane or the parasitiferous vacuolar membrane. And here we have parasite markers, either UIS4 or HSP70. And mucin 13 is co-localizing with both of those markers in both Plasmodium berghei and in Plasmodium vivax that we obtained from patients in the Peruvian Amazon. And so it seems like this marker is, this co-localization is true for both human and rodent model parasites. And then looking at the timing and the kinetics of this, the Plasmodium berghei liver stage infection cycle is about 48 hours. And so you actually don't see the upregulation or the co-localization of mucin 13 with the Plasmodium berghei parasite until quite late into the infection, which is consistent with our RNA-seq data. So at 24 hours, we saw no real upregulation. It was only at 48 hours that you ended up with about a 50-fold upregulation. And so you can see that there as well, is that it's not really visible at 2 hours post-infection. You can sort of make out the parasite, but the level does not rise above that of the neighboring host cell expression until about 36 hours, to where the signal is quite prominent moving forward. And so just looking at this and this co-localization, the one concern we have, and is always a concern with this, is how did we know that this antibody was actually binding specifically to mucin 13 and it wasn't just co-localized, or it wasn't just cross-reacting with something, a protein in the parasite. And to do that, I'll show 2 means by which we've attempted to validate that. The first was doing a knockdown of host mucin 13. Now, obviously, CRISPR is all the rage, and in Plasmodium, it's almost become ubiquitous to use. And so that's actually why I decided to go with an older school, more traditional shRNA knockdown, is Plasmodium has never been shown to have any ability to perform shRNA knockdowns. So I thought doing a knockdown instead of a knockout, there was almost no chance of having cross-reactivity. So that if we did see a decrease in the protein abundance and the signal, it almost had to be due to a host protein. And we do see a downregulation of with the knockdown here, we see about a 70% knockdown. And you do see actually that the signal for mucin 13, both in the neighboring host cells as you'd expect and with the parasite, is almost completely removed. And related to this, because we are also trying to see, in addition to just biomarkers, whether these could be actual factors involved in parasite development, we see that using a P. berghei model that expresses luciferase, when you knock down mucin 13, you actually see about a 50%, 40 to 50% reduction in the number of parasites developing in the liver stage, suggesting that mucin 13 might actually play a role in liver stage development. The second form of validation we did was fairly straightforward. We just used a different antibody binding to a completely different region of the protein. Here you can see the position of the first antibody near the transmembrane domain. We chose a second antibody at the opposite end of the protein near the glycosylation domains, and you see a nearly identical pattern of co-localization here using HSP70 or mucin 13. And you can see that it co-localizes so that mucin 13 can actually serve as a marker of parasite infection regardless of which antibody we chose. And so the final thing, if we wanted to have a good biomarker moving forward, was whether you could actually use this in a quantitative way, whether you could actually predict parasitemia or drug response using this marker. And we did see that we could. Here, just measuring parasitemia of infection in either Plasmodium berghei, you can see that using high-content imaging For HSP70 or human mucin 13, you get nearly identical infection rates of about 1.2% for those infections. Or in Plasmodium vivax, in an in vitro infection of HC04 cells, we again see nearly equal infections, though the infection rate is quite low at about 0.004%, suggesting that the marker actually can be equally as sensitive as HSP70. And you can, in fact, use this mucin 13 marker for drug response as well. As you can see, again, using a high-content imaging assay, we got nearly identical IC50s for atovaquone or purimycin when we measured based on positive events for HSP70 signal or for human mucin 13, suggesting that mucin 13 actually, as a human marker, is usable in terms of assays going forward and is quantifiable. And so just to summarize, from our RNA-seq data, we think we've identified a host biomarker of multi-species Plasmodium infection, at least for Plasmodium vivax and Plasmodium berghei, which is this mucosal immunity protein mucin 13, which is surprising to us, but it seems to be true given that mucosal immunity did not appear before this to play a role in malaria development. Its mucin 13 co-localizes specifically with the parasite later during the infection cycle within hepatocytes, and mucin 13 overexpression appears to be involved in facilitating late-stage parasite development. And I have two hypotheses for this. In terms of the in vitro setting, mucin 13 has been shown in cancer cells to affect host cell apoptosis, to actually delay apoptosis. And so it's possible the parasite is inducing the upregulation of mucin 13 to delay hepatocyte apoptosis. For as the parasite develops, the host hepatocyte expands about 4 times in volume, and so that's obviously a fairly traumatic event for the cell. And so you would want to delay apoptosis until the parasite was fully developed. And you could also imagine a scenario where coating the emerging merozoites with a host mucosal immunity protein would help it evade the immune system as it invades nearby red blood cells. And future directions, we've also made knockout cell lines of mucin 13, and this will be used to get a better sense for how it's involved in parasite development and to further validate the antibody. And we're working, and we actually might have a mucin 13 reporter line so that we could do similar RNA-seq studies or phenotypic screening in Plasmodium vivax or Plasmodium ovale moving forward to conduct similar studies. And in case other people want to hear, as I mentioned, there will be a talk in the late breaker abstracts discussing additional host biomarkers from that group of 42 genes. And at the same time, rather inconveniently, there's actually a poster discussing how these host biomarkers specifically focusing on identifying Plasmodium vivax infection in liver cells. So with that, I'd like to thank the entire Winsler lab, especially Elizabeth Winsler, Pamela Arguello-Sanchez, who's a wizard at the microscopy stuff, Jason for help with the Plasmodium vivax studies, various other helpers, the lab of Joe Vanetz for helping allow us to get access to Plasmodium Plasmodium vivax sporozoites for Peru, and the lab of Nathan Lewis for helping me analyze the transcriptomic data, and of course our funders. And with that, I'm happy to take any questions. Thank you. Yes.
It's a great story. I'm interested in what is known about the upstream regulation of mucin 13 expression and how that might be connected, how such signaling might be triggered by the Plasmodium infection in the liver cells.
So it's a bit of a mixed bag there in that the cancer literature suggests one thing for mucin 13 and the infectious disease stuff suggests other things. So there's sort of a P53 connection to this as well. We also think that there might be like a CS, that we think the parasite might be doing, expressing it through like CSP or something, leading to its upregulation, but it's not really clear to me from the literature how it's given that two very distinct fields have very distinct hypotheses on how MUC13 is upregulated.
Thanks.
Yes,
what do you know about the allelic diversity of mucin 13 polymorphisms in the human population and the geographical distribution of those polymorphisms?
I am not all that familiar. As far as I know, it's reasonably diverse in that glycosylation region. Yeah. You see a fair amount of things, but then the EGF2 and 3 domains are pretty conserved from what I've seen. But I haven't looked into that too deeply.
Might be worth looking at the geographic distribution relative to malaria infection and thinking about CRISPRing those in to see whether you can actually assess the apparent phenotypic
That's an excellent
differences.
suggestion. We've been starting to work on that. But yes.
As a biomarker, I think using the host product, you can have a potential issue. You just touch only in cancer. So if a patient with cancer, then this is induced. So how can you tell this is induction is due to virus infections or due to cancer?
So that is
malaria.
That's a very good point, and that is a concern for, especially for mucin 13, since it is upregulated in other contexts. The one thing I will say is that most of the other studies, either in Helicobacter pylori or in cancer, see about a 2- or 3-fold amplification of mucin 13, whereas we see about a 50 to 60-fold amplification. So it's possible you could design a test to look for a certain threshold, but that would be a concern going forward. You would realistically have to pair this with other things, I would think, to get an accurate sense. So it would be an additive biomarker, not a standalone, I would say.
Okay, I think we better move on.
Thank you.